Hello everyone, and thank you for attending my presentation today entitled Using a Predictive Bioinformatics Algorithm of Difficult Neoantigen Peptide Synthesis for Precision Immuno-Oncology Therapeutic Development. Today's presentation will be separated into four main sections. First, I will give a brief introduction into the world of neoantigen-based personalized immuno-oncology therapeutics. Next, I will give an overview of how neoantigen peptides are utilized during precision therapeutic development and why neoantigen peptides are so difficult to synthesize. Then, I will review GenScript's four-tier platform for synthesizing difficult neoantigen peptides, highlighting our proprietary algorithm for predicting neopeptide synthetic difficulty, before finishing up the summary and 15-minute Q&A section. As many of you will unfortunately understand firsthand or through a personal connection, cancer is the world's second leading cause of death, only behind cardiac arrest. In 2018 alone, 17 million people were diagnosed with cancer in the United States, with unfortunately 56% of them passing due to the disease. So with a 70% increase in cancer diagnosis over the past 20 years, and 9.5 million people dying from cancer in the US alone in 2018, researchers and clinicians have realized that standard methods of care, such as chemotherapy, radiation, and other one-size-fits-all treatments, are not negating the disease as quickly as cancer mutates to avoid therapeutic efficacy. Therefore, we need a new standard method of care, such as personalized immunotherapy. Immunotherapies for cancer treatment have been around for almost 10 years. However, current treatments will only target shared targets, such as oncogenes, tumor-associated antigens, or immune checkpoints. Checkpoints are receptors on the surface of T cells which, when bound by their ligand, are able to inhibit T cell-based immune response against target cells. Many cancer cells are able to avoid immune detection through expression of PD-L1, the ligand for PD-1, which will bind T cell checkpoints and inhibit immune attack. Checkpoint inhibitors are proteins which block specific immune checkpoints such as PD-1 and initiate a T cell-mediated immune response against the tumor through binding of the T cell receptor, or TCR, and the tumor-associated antigen. Clinical trials using checkpoint inhibitors have shown great potential, although the effect is limited in many cases, especially in solid tumors where checkpoint inhibitors cannot make direct contact with the tumor microenvironment. Another common immunotherapy against shared tumor-associated antigens are T cell therapies. There are many different forms of T cell therapy. However, the goal of each form is the same: to target patient or healthy donor T cells to the tumor to bind to a tumor biomarker and initiate an anti-tumor killing response. To generate T cell therapies, patients' autologous T lymphocytes or donor-matched allogeneic T cells are genetically modified to express TCRs, which bind to specific tumor-associated antigens. These T cells are then expanded in vitro and reinfused back into the patient. These TCRs can be standard TCRs, or they can be chimeric antigen receptors, better known as CARs, which on top of having a tumor-associated antigen binding domain, or TA, they contain an internal switchboard which, when bound to its target, activates the T cell to initiate an immune cascade. Clinical trials have shown great promise for these therapeutics as well. However, they also show poor efficacy in solid tumors and tumors with high mutational burden due to immune escape. Despite showing promise, one-size-fits-all or off-the-shelf style immunotherapies have shown limited efficacy in about 70% of tumors. But why? Well, there are two common ways cancer evades the immune system, as well as TA-targeted therapeutics. Those are antigen escape and weak immunogenicity. Antigen escape is a natural caveat of cancer's high mutational nature, in which shared tumor antigens are either completely or partially downregulated, allowing tumors to evade single-target therapies like checkpoint inhibitors and CAR-T. Weak immunogenicity occurs when oncogenic tumor-associated antigens are also expressed in healthy tissue, just on a lower scale, and therefore are recognized as self-antigen. Because of this, immune cells will not initiate an immune response, and cancerous tissue will remain unscathed. However, both of these common problems can be avoided by using personalized immunotherapies, as opposed to one-size-fits-all treatments. But what exactly is precision medicine? In precision medicine, individual patient tumors are biopsied and used for NGS, whole exome sequencing, RNA sequencing, and proteome identification. Through bioinformatics analysis of these sequences compared to healthy tissue, clinicians can identify treatments which will work for the specific tumor biomarkers present in that patient's tumor. There are two types of precision medicine. First is to match patient tumor biomarkers to a database of clinical and therapeutics known to be efficacious for those specific biomarkers. Second are personalized therapeutics, which are designed from scratch to target patient tumor-specific antigens known as neoantigens, the main topic of today's presentation. Neoantigens are new or novel cancer antigens which are present on the cancerous tissue but not expressed within wild-type tissue. They are expressed within individual tumors or tumor cells of a specific patient rather than being shared amongst patients suffering from one type of cancer. Neoantigens are expressed as peptides within cancerous tissue from single amino acid deletions and/or insertions known as indels. In rare instances, neoantigens can also be expressed from frameshift mutations. Because of how they are generated, neoantigens tend to be highly hydrophobic in nature, as well as having a strong tendency to aggregate. However, since they are minimally affected by antigen escape or weak immunogenicity, they make great targets for immunotherapy. There are four main reasons why neoantigens are preferred targets for personalized immunotherapies. First, normally, neoantigens are not expressed at a high enough level to be tracked by the immune system, but because they are not recognized as self-antigens like common TAs, they are highly immunogenic. Therefore, directing targeting can enhance the natural immunogenicity of neoantigens to elicit a strong anti-tumor killing immune response. Secondly, once a neoantigen forms, there is a low chance of it mutating again and leading to a therapeutic antigen escape, as it is not an important protein for oncolytic survival. Third, some neoantigen therapeutics, such as personalized cancer vaccines, tend to target multiple neoantigens simultaneously, avoiding the possibility of immune escape even if one neoantigen was to mutate and be untargetable. Lastly, each neoantigen can be matched to specific TCRs, and therefore their immune response can be closely analyzed in vitro before being used in a possible therapeutic. There are two main therapeutic types which target cancer neoantigens: personalized cancer vaccines, or PCVs, and T-cell therapies. Personalized cancer vaccines work similarly to infectious disease vaccines in that they introduce small peptide components of the biological threat you are trying to initiate an immune response against. In the case of a PCV, this will be cancer neoantigens. Since every PCV is generated for a specific patient, the first step in production will be to take a tumor biopsy. This sample will then be sent to the lab where it will be used for NGS, next-generation sequencing, RNA sequencing, and immunopeptidome identification. These sequences will be compared with sequences from healthy wild-type tissue using bioinformatics tools in order to identify the mutanome, or mutations present within cancerous tissue that are not expressed in wild-type cells. The mutanome will then be put through bioinformatics algorithms to identify the predictive immunogenicity and PCV efficacy of each individual neoantigen. The top neoantigen peptide candidates will be synthetically produced and used for in vitro functionality screening in order to confirm the predicted activity of each candidate. From these results, clinicians will first decide on the 20 or so neoantigens which they plan to use for their patient's PCV, and secondly, decide which modality they will use to introduce those neoantigens to the patient's lymphatic system. The most common modalities include neoantigen DNA, mRNA, peptide, or post-antigen presenting cells mixed with varying delivery vehicles and adjuvants. Once formalized, the vaccine will be placed directly into the patient's lymphatic system through injection in order to be processed and presented by patient APCs and initiate a T-cell-based anti-tumor killing immune response against cancerous tissue expressing those neoantigen epitopes. The second main therapeutic targeting neoantigens are personalized T-cell therapies. The main difference between personalized T-cell therapies and off-the-shelf treatments I described earlier is that they are tailored to immunogenic neoantigen on patient-specific tumors. Similar to PCVs, the first stage of personalized T-cell development is to biopsy and sequence the patient's tumor to identify the mutanome, which is put through bioinformatics algorithms to identify neoantigens are predicted to initiate a strong immune cascade when they bind to TCRs. These neoantigen candidates will be synthetically produced and screened for therapeutic efficacy in order to identify the perfect neoantigen for an individual's tumor. From there, a TCR will be engineered to express binding domains for that specific neoantigen and cloned into patient-specific, known as autologous, or healthy donor, known as allogenic T cells. Those T cells are expanded and infused into the patient in order to elicit an anti-tumor killing immune response. No matter the type of neoantigen-targeted therapeutic, researchers will require production of synthetic neoantigen peptides during 3 phases. First, neoantigen peptides are required for preclinical functionality screening of bioinformatics candidates. For T-cell therapy, these peptides will be utilized for TCR binding affinity and immunogenicity analysis, while neoantigen candidates for PCV development will be used for T-cell stimulation assays such as ELISpots, flow cytometry, and cytotoxicity assays. After a new therapeutic has been developed, it will be first tested in animals, such as mice and non-human primates, not just for efficacy testing, but mainly for safety and toxicity of novel additions to the therapeutic, such as adjuvants and delivery vehicles like lipo or nanoparticles. For this stage of development of PCVs, neoantigen peptides will be used as model vaccines to inject into the animals, as well as for post-clinical efficacy screening. During this phase, whether it be in animals or human patients, neoantigen peptides are utilized to ensure that the intervention initiated an immune response, which can be continuously used to target and kill neoantigen-expressing cancerous tissue. Despite neoantigen peptides being required for multiple stages of personalized therapeutic development, they tend to be the bottleneck for many researchers trying to produce these treatments in a timely manner due to the difficulty of neoantigen peptide synthesis. But how do we synthesize peptides? Let's review the basics of synthetic peptide synthesis. Peptides can be produced using either recombinant or synthetic technologies. Recombinant synthesis is simply expressing the peptide within a microorganism through standard plasmid transfection and peptide purification. Chemical synthesis, on the other hand, is much more involved, but basically a resin is used to anchor the first amino acid in the peptide chain, while additional amino acids are added one by one through a standard dehydration reaction between the amino and carboxyl groups of the supporting and incoming amino acid. These 2 synthetic methodologies can also be combined depending on the specific amino acid sequence which needs to be produced. At GenScript, we prefer to use synthetic peptide production because it's easier to custom design any peptide sequence, produce longer peptides through ligation of shorter fragments, and to generate large-scale peptides. While recombinant expression has limitations for incorporating unnatural amino acids or modifications, it's difficult to synthesize hydrophobic peptides, and overexpression of membrane peptides can actually harm the cell and lead to cell death. Let's dive a little deeper into chemical peptide production. Chemical peptide synthesis most often occurs by coupling the carboxyl group of the incoming amino acid to the N-terminus of the growing peptide chain. This C-to-N synthesis is opposite from protein biosynthesis. In order to accomplish this, each amino acid will go through protection of reactive groups. This ensures that when new amino acids are added to the growing chain, they bind in the C-to-N format rather than through the side chain. Once protected, the first amino acid will be linked to a resin through its C-terminal, so that new amino acids can be coupled to that amino acid's N-terminal, as shown here in step 3. Once the growing amino acid chain is finished, all of the protective groups will be removed in order to have a fully reactive peptide. This peptide will then be purified from the resin using high-performance liquid chromatography, or HPLC, as you can see through this example chromatogram in step 5. At GenScript, we use 3 different types of chemical synthesis. Liquid phase synthesis is the original methodology for synthetic peptide production, and unlike solid phase synthesis, does not require linking of the first amino acid to a resin, but instead requires an additional chemical protective group on the C-terminus of the first amino acid. Because of this synthetic technology, Each new peptide must be manually purified before addition of the next amino acid. Now, while this can be timely and costly, it also ensures extremely high purity of the final peptide. Also, because the C-terminus is not attached to a resin, it is the preferred method for any peptide requiring C-terminal modifications and is great for large yields. Solid phase synthesis is similar to liquid phase synthesis. However, instead of a protective group on the first amino acid, the C-terminal is linked to a resin. This makes addition of new amino acids easy and smooth, and therefore significantly reduces turnaround time as purification can wait until the end. Because of the technology, solid phase synthesis is preferred for long and difficult peptides rather than bulk production. The most recent advancement in peptide synthesis is through the use of automated microwave technology. Rather than manual synthesis, microwave synthesis is done entirely through a machine, and therefore can significantly reduce the turnaround time and increase the overall capacity to make larger numbers of peptides per day. In fact, GenScript's microwave technology actually doubled our capacity, so now we can chemically synthesize 18,000 peptides per month. Because this technology uses microwave energy, it is able to increase turnaround time by reducing the time required for coupling and resin separation. This also makes it great for fast synthesis of difficult shorter peptides rather than longer ones. The reason we utilize 3 different types of chemical synthesis and a combination thereof, including recombinant production, is because each peptide's intrinsic characteristics will dictate if a peptide is difficult to synthesize and why. Neoantigen peptides tend to be highly difficult to produce, and it will require all different types of synthesis in order to produce highly pure peptides at required yields. Specifically, neoantigen peptides can be difficult to synthesize because of these 5 characteristics listed here: peptide length, charge, hydrophobicity, yield, and purity. For length, neoantigens tend to either be 9 to 11 amino acids representing the neoantigen epitope, or they will be larger than 25 amino acids in order to be processed and presented by APCs on MHC class 1 and class 2. Peptides over 20 amino acids tend to be difficult to synthesize and will usually require a combination of microwave and solid phase synthesis. For peptide charge, because neoantigens are only expressed in cancer, they tend to contain charge-modifying amino acids which would not be expressed within natural tumor-associated antigens. Many synthetic steps only work with specific isoelectric points, and therefore the further from neutral a peptide is, the synthesis will be more difficult. Third, because neoantigens are produced from random amino acid indels throughout the peptide, hydrophobic side chains are bound to be introduced within the sequence. These hydrophobic side chains will lead to a stronger chance of aggregation and therefore limited solubility in water, which in turn makes purification very difficult. Neoantigen peptide synthesis can also be very difficult because each application within the various stages of therapeutic development will require completely different yields, or peptide mass, as well as purity, or percentage of target peptide within a sample. Because of the flexibility required, it can be cumbersome to find a reliable provider which is able to accommodate yields ranging from large-scale microlibraries of half of milligram to synthesis of gram level of one individual peptide. Because of this, GenScript has tailored its neoantigen peptide synthesis service to be fully customizable in order to achieve yields for any application. In terms of purity, other than flexibility required for various applications, because neoantigen peptides tend to be hydrophobic and have a tendency to aggregate, their solubility makes them difficult to purify using standard techniques. Because of this, they must be treated with extra care and go through additional processes of purification in order to reach the 98% purity required for applications like personalized cancer vaccine generation and analysis in patient-derived cell lines, as these are precious samples. In order to accomplish difficult neoantigen peptide synthesis, we here at GenScript have developed our own neoantigen peptide synthesis platform, which utilizes reliable technologies and tools to achieve a 95% success rate for difficult peptide synthesis. This platform can be divided into 4 main stages. In the first stage, every neoantigen peptide sequence which comes through our order system is analyzed by our proprietary NeoPre bioinformatics algorithm in order to determine synthetic difficulty and the best method to synthesize each peptide based on its predicted difficulty. In the second stage of GenScript's platform, those neoantigen peptides will be synthesized according to the recommendation of our NeoPre algorithm and our peptide experts, using one or a combination of our synthetic technologies, ranging from manual liquid and or solid phase, automated microwave technology, recombinant expression, or chemical ligation. In order to understand how we achieve this 95% success rate for highly difficult peptides, let's dive a little deeper into GenScript's NeoPre analytics tool. GenScript's long-standing experience in peptide synthesis has given us an opportunity to develop a tool for estimating ease of peptide synthesis process. It is necessary to evaluate the feasibility of all peptide sequences prior to synthesis. Therefore, every one of our customers' peptides go through our proprietary NeoPre bioinformatics algorithm to determine synthesis difficulty. The first step in using this tool is to copy the neoantigen peptide sequence into the algorithm shown here. If this is the customer-facing tool, then once you hit submit, you will see a response indicating how difficult the synthesis of the peptide will be. If this tool is being used internally, the readout will be much more involved and highlight the individual characteristics that are causing difficult synthesis as well as the recommended synthetic technology. This slide shows an example of what the readout of the internal algorithm would look like. The algorithm would analyze the individual peptide sequences and convert its intrinsic characteristics to numbers which correspond to how difficult the peptide synthesis will be due to that specific characteristics. The 6 characteristics that NeoPre analyzes are tendency of the peptide to aggregate, represented by Na4VSS, isoelectric point, shown as Pi, hydrophobicity, sequence length, yield, and purity. Amino acids are shown in red during the system review to indicate which ones have strong tendency to aggregate. For aggregation, less than 0 is considered simple, 0 to 20 is considered middle, and over 20 is considered hard, while larger hydrophobicity numbers indicate more difficult sequences. Once the algorithm has assessed each peptide's difficulty, it will determine the predicted difficulty of synthesizing each peptide as well as recommend the best means of successfully synthesizing each individual peptide. Once the GenScript peptide expert reviews the results, they will then adjust the synthetic methodology as needed based off GenScript's 15 years of peptide synthesis. NeoPre is able to analyze the numerical values assigned to individual characteristics of each peptide in order to predict the synthetic difficulty. Overall, GenScript has used NeoPre for 171 peptides for this specific case study. Of those, 71 were shown to be difficult to synthesize. Of the 71 difficult peptides, 61 were actually difficult to synthesize, while 10 were actually easier to synthesize than originally predicted. Digging deeper, 100 peptides were shown to be easy to synthesize, with 78 of them accurately predicted and 22 of them actually difficult to synthesize. With this tool, what's important to understand is that those 83 total difficult peptides were able to be synthesized despite their high hydrophobicity, long lengths, and tendency to aggregate. Since using NeoPre, GenScript has actually been able to synthesize 2,158 out of 2,267 peptides ordered, giving us an overall success rate of 95.15%, 37.5% of which were delivered using GenScript's Rush peptide service within 5 to 6 days. Neoantigen peptides tend to either be focused on epitopes presented on MHC1 and MHC2, which range from 9 to 15 amino acids, or they tend to be larger, over 20 amino acids. For 9 to 15 amino acids, GenScript synthesized 1,835 out of 1,869 peptides ordered, giving an overall success rate of 98.18%, with the majority of these peptides being deemed as difficult. Synthesizing the larger peptides, over 20 amino acids, always tend to be more difficult and require more attention in synthetic methodology and take slightly longer. Because of this, GenScript has synthesized 85.31% of ordered larger peptides. After each neoantigen peptide has been synthesized using our NeoPre and synthetic technologies, they will go through stringent quality control based on individual customer and application needs. All GenScript peptides undergo total quality management testing, including mass spectrometry and analytical HPLC, high-performance liquid chromatography analysis, throughout the peptide synthesis and purification process through our trademarked AccuPep quality control service. In addition to the standard COA document you would receive, GenScript offers an additional 12 quality control services, including our endotoxin analysis and removal service. Endotoxins are lipopolysaccharides, which are major components of the cell walls of gram-negative bacteria, which can be introduced into custom peptide preparations during synthesis. More importantly, they can stimulate a plethora of unwanted immune reactions in B cells, macrophages, as well as stimulate T cell expansion even at the lowest concentration. To enhance your experimental results, GenScript offers custom peptides with guaranteed endotoxin control service to reduce endotoxin levels at less than 0.01 EU per microgram, which is ideal for most cellular assays, especially on primary cell lines. We also offer guaranteed TFA measurements and removal. TFA, or trifluoroacetic acid, is a strong acid which is commonly used to cleave synthesized peptides from solid phase resins, and is also used to improve HPLC performance in the peptide purification step. Although TFA is quite important, residual TFA in custom peptides can cause inexplicable discrepancies in subsequent assay data, which is why at GenScript we can guarantee TFA values of less than 1% in exchange for either acetate or formate. The last step of GenScript's neoantigen peptide synthesis platform is peptide optimization, in which we can design 7 different types of peptide libraries in order to narrow down your perfect neoantigen sequence. The most popular peptide library design services for neoantigen peptides are GenScript's alanine scanning, overlapping, and T-cell truncation libraries. In an alanine scanning library, each amino acid will sequentially be replaced with an alanine. In turn, this will allow the end user to determine the most important individual amino acids to peptide function. As opposed to an alanine scanning library, which replaces individual amino acids, an overlapping library simply deletes amino acids over a certain threshold. Because of this, it is great for identifying where a neoantigen epitope is within an entire neoantigen peptide sequence. T-cell truncation libraries are similar to overlapping libraries, except you can decide on multiple epitope lengths to put into one pool in order to tease out the perfect length of your neoantigen or TCR. I know I have gone over a lot of information about neoantigen peptides and how they are used in personalized cancer medicine. So now I would like to summarize the key takeaways from today's presentation. First, neoantigens are new or novel cancer antigens which are present on cancerous tissue but not expressed within wild-type tissue. They are expressed within individual tumors or tumor cells of a specific patient rather than being shared amongst patients suffering from one type of cancer. Second, normally neoantigens are not expressed at a high enough level to be tracked by the immune system, but because they are not recognized as self-antigens like common tumor-associated antigens, they are highly immunogenic. Third, neoantigen therapeutics can be either personalized cancer vaccines or personalized T-cell therapies. Both of these therapeutics require neoantigen peptides for various stages of functionality and efficacy screening. Fourth, GenScript's NeoPre bioinformatics algorithm allows GenScript to synthesize highly hydrophobic difficult peptides by analyzing the individual amino acid sequence for hydrophobicity, length, aggregation, charge, yield, and purity to identify synthetic difficulty and the technology to synthesize each peptide. And lastly, GenScript's neoantigen peptide services offer a reliable synthesis by being a trustworthy partner, having a high success rate for difficult peptide synthesis and flexible services for all precision therapeutic applications. I want to thank you for attending my presentation today, and I am happy to answer any questions you may have. But while you are typing your questions into the chat box, I would quickly like to mention that GenScript's neoantigen peptide synthesis service produces reliable neoantigen peptides for precision therapeutic discovery. This is because at GenScript, we have the technical means and experience to synthesize large numbers of challenging neoantigen peptides and provide tailored optimization tools for your downstream applications. We are able to accomplish this by providing a trustworthy user experience demonstrated through our work with over 50 of the world's leading immuno-oncology organizations, at 95% success rate for difficult neoantigen peptide synthesis, and our extreme service flexibility, putting our customers in the driver's seat of their entire project, no matter what the application. Again, thank you for your attention, and I am looking forward to answering your questions. Thank you.